Hi everyone, welcome back to Synthetic Biology 1. Today we will be measuring DNA concentration by absorption. The principle of this protocol is that DNA absorbs UV light. Actually, different nucleotides absorb wavelengths at a little bit different wavelengths, but the overall peak is around 260 nanometers. We will be using a tabletop spectrophotometer, commonly named nanodrop. So first, I will have to run a blank using the same solution as you used for dilution of the DNA. In this case, it's water. So I will put 1.5 microliters of water and click blank. Okay, now that the blank is made, I can load the sample. But before that, I will wipe the measuring area with a clean tissue, and now I can load 1.5 microliters of the sample. Make sure that there's no bubbles. Okay, and so you can see that there's a prominent peak at 260 nanometers, which corresponds to the DNA. And the concentration that is measured at this peak is around 200 nanograms per microliters. However, it is also important to look at peaks around 230 and 280 nanometers, because this corresponds to wavelengths at which possible impurities will absorb. In this case, there's no peak at these wavelengths, meaning that the DNA is pure and the concentration corresponds to the concentration of the DNA. However, one common mistake is to measure DNA concentration from the unpurified PCR samples. Let me show you what kind of results it will give. So I will load 1.5 microliters of an non-purified PCR sample and click measure. So you can see that not only there's a peak at 230 nanometers, but also the main peak is shifted from 260 towards 280 nanometers. So this corresponds to impurities such as proteins, salts, and other components of the mix. Another point is that even though the concentration is measured, you cannot take this concentration as the actual concentration of your amplified DNA in the mix, because not only amplified DNA strands will absorb at this wavelength, but also unused DNTPs, which means that this concentration does not correspond to the concentration of your PCR product. So our DNA sample was concentrated and pure, and so the measurement was successful. Until next time, stay concentrated.